Yeah, please.
Are you gonna kind of like go over that one blast exercise you posted on Google Classroom? Because I have, struggling with that one.
Okay, so the big exercise. I will eventually, but not before the exam, which means it's not going to be on the exam. Yeah. So thank you for asking. It's a good point to clarify. So it is important to know how to do all of that, but there's not enough time to practice it sufficiently before the exam. Yeah. We will get there eventually. Other questions? At some point today, we're going to stop content for the exam, and then Friday will be a review session as usual. Monday will be the exam. So Sunday night, this activity was due. Short post to the Canvas discussion board about this fact is, so an orphan gene, by the way, is a gene that is found in just one species, or maybe a small number of species, closely related species. So is the orphan gene NCYM found only in humans and chimpanzees? This is directly related to what we're working on right now in class, not only the concept of the gene, but on how you find genes and whether or not you can how you would detect it if a gene is present in a species. So what technique could we use to do this? If we have the DNA sequence of the human NCYM gene, what could we do to find out if it's present in other species? What did these authors do? Blast, right? They had a blast. And so specifically, and this is content, this is vocabulary we're going to talk about today, because most of you who answered this prompt and posted on Canvas had the same citation. This is the one I found as well. This is a paragraph from this citation. We searched for paralogs and orthologs, and that's what we need to define here, because that can be a little bit misleading. Using BLAST, and the e-value threshold is relevant but not relevant to us today. We did not find any paralogs, but identified orthologs in what species? Baboons, chimpanzees, and chimpanzees. So is this gene only in humans and chimpanzees? And baboons, at least as far as these authors could tell. So in this paper, they do say that they found evidence. That doesn't mean that this gene is there. They found evidence of it in at least 3 different species: chimpanzees, baboons, and humans. This is a little bit misleading. And of course, you didn't get it wrong if you answered, yes, this fact is true, and cited this paper, because they did something here that's a little bit misleading. They then said, and a lot of you said this, we focused on the NCYM gene of the hominidae, which is a group of primates. And a lot of you found other papers, publications, that cited this study that said this gene is only found in humans and chimpanzees, because those are the organisms that are in that group of animals. In other words, here is the family tree of primates. We've got humans, and then we've got the non-human primates: chimpanzees, gorillas, orangutans, gibbons. This is kind of like a family tree except upside down. So the common ancestor is here at the bottom, and then splits in the family tree. Speciation events where different species evolved, occurred. So this is why humans, for example, were close relatives with chimpanzees and bonobos, 2 different species that are both part of the genus Pan. Of course, we're the only member of Homo. We're the only currently existing species in the Homo genus anyway. We're Homo sapiens. All the other ones went extinct. Does anybody know another Homo species? Homo naledi? That sounds familiar. There's Homo neanderthalensis. Neanderthals were a different species of humans. They went extinct. Sapiens is the only currently existing species in that genus, Homo. So the point being, when those authors wrote, we focused on the NCYM gene in the hominidae, that meant that necessarily they were focusing on which species? Humans, chimps, gorillas, and orangutans. Those are the 4 different types of species that are found in that group called the Hominidae. So what about baboon? They found evidence for this gene in the baboons. Why didn't they talk about it in that paper? Say it louder. Yeah, because are baboons in the group that they said we focused then on studying NCYM gene in this group? No, they're not in that group. So they found evidence for it here, but the authors were saying, we just didn't study that. Pay no attention to this gene over here. We're going to focus on that gene in this group of species, which is just humans and chimps. They said, or they didn't say they found evidence for this gene in gorillas and orangutans. They just found evidence for the gene in humans and chimpanzees and bonobos. But we're not going to talk about them right now. So there's evidence for the gene in other species besides humans and chimps. It's easy to see, I think, how you could come to the conclusion if you just read that paragraph, and I think that's what the authors of the National Geographic article did, was read that paragraph and say, oh, they just found it in humans and chimps, that's what they studied. But there's evidence for this gene existing outside of that group.
What's the difference between a paralog and an ortholog?
Yes. Great leading question. So, I'll make one point first, but thank you for bringing that up. This, by the way, is another example of NCYM, the gene. If you look it up in NCBI, they actually call it NYCN, opposite strand. That's the formal name of the gene, I guess. It's a non-coding RNA. So it's another one of these genes where the gene's role is not to be translated into protein. The purpose of this gene is just to be transcribed into an RNA. Just another example of one gene that doesn't get translated. So talking about genes, some definitions: orthologs and paralogs. This also won't be on the test, but it will be on the final. So it's not a get away, go home free card sort of situation. It's just a take notes now, but don't focus on this for studying. So here's another family tree, and all phylogenetic trees are usually drawn like this, where the time runs from, well, depends on how the tree is drawn. The tips of the tree are drawn at the time of present day. So now. In this case, what this diagram represents is one group of organisms, a population or a lineage, that exists through times from really long ago to more recently in time, and then at some point that population splits into 2 groups. A group of organisms splits into 2 groups of organisms. That might be, in this case, we'll call that a speciation event, where one species becomes 2 species. And you'll learn more about this when you get to Biology 105, evolution, that's not actually an instantaneous point in time where all of a sudden one group of organisms becomes 2 totally separate species. That is just the best way we can draw the process, as just a one point in time split. Not necessarily true. So let's say, just for the sake of example, that we're talking about something familiar like mice and rats. So closely related animal species that evolved from a common ancestor. So they have a common ancestor long ago, and then at some point, as time proceeded, as the years passed by, that species split into 2 and evolved into the present-day mouse and present-day rat. Do the mice and the rats have the same genes? What do you think? What was that?
Partially.
Partially. That's a great answer. Mostly yes, because they evolved from a common ancestor. So they share a lot of the same genes. It's like humans and chimpanzees. We have a lot of the same genes. We're close relatives. But are we chimpanzees, or are chimpanzees humans? No. And that's because there are genetic differences between different species. That's true of mouse and rat. So let's invent a hypothetical gene. We're going to watch what happens with gene A. Single gene. Call it gene A. way back in time, before mice and rat became different species, that ancestor, whatever organism it was, pre-mouse rat, ancient rat, had gene A. Will mice and rats, do you expect mice and rats will also, at present day, have gene A? Sure. I'm seeing nodding heads. I'm not hearing voices, but that's fine. So mice and rats, yeah, still have gene A. They inherited it from their ancestor. It's on a chromosome. They both inherit it. But what happens if gene A, and this does happen regularly, duplicates? So at random, sometimes genes sitting on our chromosomes, duplicate. It means that all of a sudden, in one organism, that chromosome that contains the gene maybe has 2 copies of that gene. We're not talking about sister chromatids. We're just talking about one piece of DNA has 2 versions of the gene. So at this point in time, after gene A duplicates, so let's say it's sitting on a chromosome, we'll call it chromosome 1 just for fun, little diagram of a chromosome down there at the bottom. Originally, there was just gene A on chromosome 1 back in time in the ancestral population. After duplication, we have to be able to keep track of the 2 gene A's. As soon as the copy, it's like copy and paste on your device. DNA sequence of gene A, copy and paste it onto the same chromosome. That's how gene duplications work. Not getting into the mechanistic details of how duplications happen. We can talk about that later if you want. So now there are 2 copies of gene A. How are we going to keep track of the 2 copies? We need some nomenclature. So what do you want to call? We can't call them both A because they're 2 different genes. So what do you want to call them?
A prime.
Okay, A and A prime?
Yeah.
Okay. So we've got gene A and we've got gene A prime. So if the gene duplication happens up here on the tree, that means at that point in time, there are now 2 genes, A and A prime. Which means that if nothing else happens, if we don't invent any other weirdness happening during evolution, that mice have genes A and A prime, and rats also have genes A and A prime at present day. This is how new genes are formed. Genes duplicate sometimes. New genes are formed this way. A gene duplicates, and then the copy of the gene accumulates mutations, SNPs, deletions, insertions, microsatellites, that make those 2 versions of the gene, A and A prime, different at the sequence level. And that's a really important thing to keep in mind. So we could invent sequences for A and A prime. Let's see, A was ATAC, and A prime is, make one nucleotide change to that. ATAC, A prime, ATAG. So they have some evidence of being the same gene, and that most of the sequence of these 2 genes is the same, and that's because if A was the sequence ATAC, after duplication, those 2 genes would have been the same sequence. Immediately after duplication, they would have been the same. You'd have 2 genes A with the same sequence on the chromosome, which means that something happens during the evolution of these species. As time passes and generation after generation of DNA replication, random mutations are inserted into genes. So let's say that mouse has a different mutation that happens to it. This happens, just inventing things. And let's say that mutation is to the first nucleotide. So instead of A for those genes, we get a G, for example. So that means present mouse sequence after that mutation of A to G, the sequence becomes GTAC for A and GTAG for A prime. To summarize, because now you're lost and your eyes are fogging over and wondering what the hell's with all these mutations. Quick summary. What's happened are there two different types of mutations have occurred. One type of mutation, well, they're the same types. They're all single nucleotide polymorphisms. They're all single base pair changes. But one type of mutation, the C versus the G at the fourth position, distinguishes the two versions of the same gene. So we can tell A from A prime based on whether it's got a C or a G at the last position. And that's true both in rats and in mice. The other difference that has occurred is that if you looked at the first position, based on this mutation, where that first nucleotide A in both copies of the gene mutated to become G, if you look at the first nucleotide of the genes that are in mice, it's G. If you look at the first nucleotide in rats, it's A. So if you wanted to know if this particular, if you wanted to know if this gene, if I just gave you the sequence, 4 nucleotides, one of these genes, and I said, is it from mouse or rat? How would you know? There's a species-specific mutation here. Mice have which nucleotide? Which nucleotide in that 4 nucleotide sequence gives us information about which species it's in?
It's the first nucleotide.
Yes, this G, right? Every mouse gene, because this mutation that caused that G happened only in the ancestor leading to mice, all mice have that mutation, but no rats do. So that first nucleotide then lets you distinguish different species because that mutation happened in the common ancestor of one species, but not in the other species. And if you wanted to tell A from A prime, if you wanted to distinguish within a species, which one is A and which one is A prime, you look at the last nucleotide. And that's true both for mice. A has a C at the last nucleotide in mice, and A also has a C at the last nucleotide in rats, where A prime in both species has a G at the last position. It's very hard to see how this could be useful without more expertise in genetics, which is fine. We'll get there eventually. But these are the definitions of orthologs and paralogs. It's just a molecular basis of the definition. So ortholog, finally to get to the point. An ortholog is the same gene in 2 different species. What I mean by that is we have 2 species up here, mice and rats, and we have 2 genes, A and A prime. So what are orthologs? The 2 A's are orthologs. It's the same gene in 2 species. There's gene A in mice, there's gene A in rats. That's the definition of an ortholog. It's like saying the RNA polymerase in mice and RNA polymerase in rats. RNA polymerase in rats, RNA polymerase in humans. They're all orthologs. It's the same gene. It's the gene that does the same function in multiple species. Paralogs are different versions of the same gene in the same individual. So in this case, the rat A and A prime down here in the bottom right are paralogs. Those 2 genes, A and A prime, share an ancestral gene way back here on the tree, at the top of the tree. Right? There was once one gene A that duplicated and became A and A prime. So those 2 genes have that gene ancestor, and they're paralogs. The same gene within the same species, paralogs. Same gene, different species, orthologs. So when those authors said we were looking for orthologs of NCYM in different species, they meant they were looking for the same gene, NCYM, in different species. To get this definition, maybe the most important thing to talk about is A prime in mice an ortholog of A in rats? The answer is no. The reason why is because A prime is a different gene. That's why we called it A prime. It's a different piece of DNA that encodes a gene that's similar to A but not identical to A. So A and A prime in mice, for example, and A in rats are not orthologs. They're not the same gene. They don't have the same sequence. So we'll have more practice, and this is why this is not on the exam either. We'll have more practice with this concept as we go as well. So here's applied genetics then. If you wanted to know if if you wanted to do the exact same thing these authors did, or if I asked you to do the same thing these authors did, you could go look up the DNA sequence of this messenger RNA molecule. NR161162 is the accession number. You could get the FASTA sequence of that gene, hopefully. If you don't know how to do that, come talk to me, we'll work on it. You get the sequence of that gene, and then you'd blast it. You'd find out what other sequences in the database, in the BLAST database, look similar to NCYM, this gene. And that's exactly what these authors did. They just didn't show us this process. But to come up with this paper where they said we found evidence of it in chimpanzees and, what was it, pygmy chimpanzees too? And bonobos. So here are the top BLAST results. What species are they in? We've got human, top down, human. The next one, some human cell line. Is there anything in here that's not the three we knew about? Humans, chimps, bonobos.
There's one that says gorilla, gorilla, gorilla.
Zip. Yeah, genus gorilla, species gorilla, subspecies gorilla. There's only one species in gorilla, but anyway, don't get me started. Right, so there's evidence for this gene, this DNA sequence being in gorillas. The authors didn't tell us that. And they might not have known it at the time because the gorilla genome sequence might not have been in the database at that point. So they might have done, as they wrote in the paper, they might have done a BLAST search with the NCYM gene. But if there's no genome sequence there of other primates, then you're not going to find matches to the gene you're looking for. And so we also find things like Ceracebus 8. I don't know what species that is. I'm guessing Cebus capucinus is the capuchin monkey. Those are the cool ones. I think, are those the ones that hang out in the hot water baths in Japan, like hot springs? There's some cool Google it. Oh, are those macaques? Are there macaques on here, actually? Doesn't look like macaque shows up here. Right, so there are a lot of different primate species in this list, which suggests that this gene is in more than just humans and chimpanzees. That doesn't mean anything about what the gene does. It probably still does the role that the authors in this paper said it does. It just means that you have to be really careful when you make conclusions about interpretations and analyses just based on information you have now. Well, let's see, I have to get off my soapbox at some point, but this is perhaps one of the most important, as a scientist, this is perhaps one of the most important points you can take with you, which I didn't learn until I was in grad school or after I got my PhD or something. But it's a really important, simple principle to guide you. This happened to these authors. Can somebody explain why the absence of evidence is not the same thing as evidence of absence? That is, these authors looked for the NCYM sequence, and they said, we only found it in chimpanzees and not orangutans.
Well, it's also like that same point you brought up that maybe gorillas showed up because when they blasted at the time, gorilla wasn't there. So there's just like a lack of information, so you can't say for sure if something is true or not if you don't know it.
Right. In other words, there's this thing called acquisition bias. We only know about the things that people have already studied. We only have information about stuff that has already been reported in the literature, sequences put in the database. So looking and not finding anything is not proof of anything, in other words. We can only, and this goes along with perhaps a broader point in the scientific method, which is that we can never prove hypotheses, which you've probably heard at some point. We can only disprove hypotheses. We can only find evidence that disproves a hypothesis, but we'll never collect any evidence that proves a hypothesis because of this principle. You could look for it all you want, but until you find evidence of something being true, you can't prove that it's true. It's a horrible philosophical circle. And that's why we can only disprove hypotheses. So all of science, and once you get to Biology 104, for those of you that are going there, if you're not already doing independent study research, you will find out all about how disproving hypotheses is horrible and boring, but that's how we make advances in science, is by creating hypotheses and testing them and disproving them. It's sort of philosophically horrible, but that's how science works. Any questions? Even though I came in late, I felt like that was a lot of talking. So maybe we can have you work together, check with a neighbor, if it's a neighbor you haven't already worked with on this exercise. Check your work with somebody near you that you haven't already collaborated with, and then let's come back together. This is the last thing we're doing for the day, and a few things to wrap up, announcements at the end, especially related to the exam. But this leads us into the next topic. So 5 minutes or less. Let's see what answers you can come up with. Did anybody get anything different on C? When you get there, no rush. We're gonna have to sit here until we get another answer for C, because something doesn't look right to me, but I'm not sure what. So we got matches. GU.
I think it looks right.
Is there another way to do the alignment for C? Yes, sir. Did I miss one? Was there supposed to be a C right here? Right there? Okay. What's another way to do the alignment in C? Thank you for pointing that out, by the way. Yeah.
You can put the G right next to the COA
Right. So that G could match in one of two spots. It can either be with the letters on the left or the letters on the right. Which one is best? Nobody knows. It's one or the other. I happen to know it's the one where the arrow is pointing now because there's some pattern that you're supposed to see in all of these alignments. So I'm going to draw that G right there. So if we do that, I'm about to crash my tablet, so this is always fun. What's common to all of the alignments we've created in terms of the sequence that's in the intron, or the bit that's missing from the RNA molecule that's present in the DNA molecule? There's a pattern in the nucleotides that are taken out. Pardon that? Pardon that. What's that? Pardon me. What was the pattern? I heard it was right. They all start with GT, and they all end with AG. So in optimally aligned, and those are patterns that we're going to see in eukaryotic introns, is that every intron starts with a GT and ends in an AG. That's a signature you can look for when you're looking at DNA to figure out where introns are. Of course, GTs and AGs happen all the time. But it's a hallmark of an intron. In the DNA, there will be a G and a T at the start and an A and a G at the 3' end, that you don't see that sequence represented in the messenger RNA molecule. So to recap, please don't worry about orthologs and paralogs for the test. We'll work on them again after the test. Friday, we've got the review session. Please come ask questions. And sometime during class today, I believe the study guide got distributed on Google Classroom, so you've all got that now. As usual, I'll post the key by Friday, so you can take a look at the key, compare your answers to it. Actually, I'll just post it today. Why would I wait until Friday? So here is the other version of the study guide. I haven't done this before, but I was trying to figure out what we had covered since the last exam so I could write questions, so I figured I'd give this to you as well, because these are the topics we've covered since the last exam. So those are the things we've done, the case studies we've looked at, those are things to study. And it's all on the PDF, so you've got that. One last thing, if you'll spare me a couple of minutes, you might be interested in this. You can read it yourself, of course. At this point in the semester, we've had 25 of the 45 exam points assigned. That means there are 20 more points of exams left. 15 for the third exam, 5 for the final. I've only assigned so far 17 of the 35 available points for exercises, meaning 18 points are going to be assigned between now and the end of this semester. And attendance. I've used up most of those points already, so there'll be 6 more Socrative activities that are 4 points during the rest of the semester. That means that at this point, you have the possibility of having earned 56 out of the 100 points, which means if you've got 100% at this point, you've got 56 out of 56, you've got a solid C already in the bag. It's all about accumulating points. What this really means is that bottom point. There are more than 2 letter grades worth of points left to assign between now and the end of the term. So if you're an A now, keep it up. But you need to earn at least 80 total points to get to an A. 80% or higher is an A, 60% to 80% is a B, and so forth. So if you've already got 56 points now, that's already earned you a C. You can only add points and get a higher grade as the term progresses. So the real point here is that if you've only got, let's say you've got 20 points now. 20 out of 56 is probably D range. Have to do the math. But with 44 more points, if you earned all of those, you can still earn a B in the class. In other words, there's a lot of points left in the term. Don't give up now. If you've been doing good all along, there's no reason to think you're going to do worse the rest of the semester. You should be fine. Keep it up. If you're despairing, don't despair, because most of the points that are left, half of them are exams, half of them or more are coming to class, doing the exercises, turning them in, and attending, trying, practicing. So I encourage you to do that.
Are all of the mistakes reflected on
Yeah, all of these scores are current on Blackboard, Canvas. Yeah.